Steroid hormones, being lipids, do not dissolve well in an aqueous medium. Therefore, to move in circulation, they have to be bound to a water-soluble molecule. These are special molecules produced in the liver called steroid-binding globulins. Steroids are released from an endocrine secreting cell into the interstitial space, and eventually they enter into circulation. Once in the bloodstream, they bind to their respective steroid-binding globulin and they are transported throughout the body. When they reach their target tissue, they dissociate from the carrier protein and leave circulation through migration between endothelial cells. Then, being lipids, they cross the cell membrane to reach the inside of the target tissue. Within the cell, they can either bind to a cytoplasmic receptor and then move into the nucleus, or, what is most common, move directly into the nucleus where they bind a nuclear receptor, forming an inactivated steroid receptor complex. This complex then releases a small protein called a heat shock protein. Once released, the steroid receptor complex changes its conformation, becoming an activated steroid receptor complex. Being activated means that the complex can now bind a special section of the DNA called the hormone response element. Upon binding to the DNA, the process of transcription of a specific messenger RNA sequence commences. The newly created messenger RNA then moves to the outside of the nucleus, where it will encode for a new protein in the process of translation. These new proteins will then carry out the effect that the steroid hormone was meant to trigger in this given cell.